the challenge with just counting and following autophagosomes is that's just a vesicle in its own right, and we think it relates to how much autophagic activity there is. But really, if we want to upregulate autophagy to clear aggregate-prone proteins, then we want to be labeling those aggregate-prone proteins and following their clearance. So recently, we developed a technique whereby you can tag an autophagic substrate, so one of the proteins which accumulates in neurodegeneration. So we chose to label tau protein in transgenic zebrafish. And instead of labeling them with a standard single fluorophore, we used one of these newer fluorescent proteins called dendra. It is green in its standard fluorescent condition, but we can photoconvert it. So with a very defined flash of UV light, we can convert the green fluorescent protein to red in a single cell. And we do this in the zebrafish because we can image and we can make transgenics very easily. So within a single neuron in the living zebrafish larvae, we photoconvert this dendra-tagged tau protein. So that gives us a steady state pool of the tau protein. And with sequential imaging, we can measure how much that pool disappears over time. So the beauty of this is, as people who've done old-fashioned sort of pulse chase biochemical experiments will know, is that you're often confounded in these studies because new protein is always being made. In this case, the new protein that's made is going to be green fluorescence, whereas at this point and with this flash of UV light, you have a steady state pool. You're not going to make any more red fluorescent protein because you've only photoconverted what's there at the time. So you can measure its clearance kinetics. And then this is amenable to manipulating autophagy with autophagy upregulating drugs, or looking at genetic modifiers and see whether they enhance the clearance, and also manipulating the proteasome. So we have an autophagic substrate, which is a neurodegeneration disease-causing protein, and we can play around with how well it's cleared in vivo in neurons.